Next presenter is Dr. Kirk Druey, who is the chief of a molecular signal transduction section at the National Institute of Allergy and Infectious Disease. And he'll be talking about respiratory epithelial interaction.
So thank you to David and the organizers for inviting me to speak here today. The title of my talk is A Fungal Protease Allergen Provokes Airway Hyper-Responsiveness in Asthma. And since we have a little bit of time, I wanted to give a quick aside. When I was an undergraduate at Stanford more than 40 years ago, Dr. Rick Moss gave me my first opportunity to do research. And we were studying fungal asthma, so thank you for that. And the takeaway, especially for the trainees and for all of us really, is to enjoy every research experience because you never know where your career is going to go. So with that, I wanted to talk a little bit about asthma and give an introduction. Obviously, it's a very pervasive disease that affects more than 300 million people worldwide. But we now appreciate this as a clinical syndrome that can be defined by reversible airway obstruction. But despite that, we now understand that the underlying molecular pathways leading to this common phenotype can be very diverse. And these have been called endotypes. In the last 5 to 10 years, really, this field has exploded, and many of these endotypes or differential pathways leading to the asthmatic phenotype have only been partially characterized. And about somewhere between 5 and 10% of patients have severe treatment-resistant disease, and nearly half of those patients are sensitized or have allergy to common ubiquitous molds such as Aspergillus fumigatus. And as you heard in one of the other seminars this morning, this has been termed a distinct endotype called severe asthma with fungal sensitization, or simply fungal asthma. This graphic contrasts the healthy and asthmatic airway. And nearly 200 years ago, when Henry Hyde Salter first described this disorder, he characterized that it was one characterized by exaggerated constriction of the airways, and for a lot of the time since then, treatments were squarely focused on combating this bronchospasm with bronchodilators. First coffee, and then other methylxanthines such as theophylline, and more recently beta-adrenergic agonists. And it's really been only in the last 40 years or so that we now understand that asthma is a state of chronic lung inflammation, specifically one characterized by type 2 immune responses. And this can lead to a number of abnormalities, including eosinophilic infiltration of the airways, airway remodeling, including collagen deposition, epithelial barrier damage, airway smooth muscle hypertrophy, and excessive mucus production. And all of these make critical contributions to the obstruction that we see in this disease. And accordingly, the treatments are now more focused on combating this chronic inflammation, initially with corticosteroids and more recently with biologics that target type 2 cytokines such as IL-4, 5, and 13. So our preclinical research program in this area focuses on the central question of how do airways, specifically the target cell in asthma, airway smooth muscle, how does it become hyper-responsive in asthma to bronchoconstrictors? And this is a central feature of asthma, so-called airway hyper-responsiveness, or AHR. And certainly one can't overestimate the number of inflammatory mediators that are present in the lung that have direct effects on airway smooth muscle contraction. Mediators such as IL-13, TNF, mast cell-derived products, including leukotrienes and histamine, and all of these can induce bronchoconstriction. But targeting any one of these mediators, with a few notable exceptions, has a limited impact on clinical symptoms. And so we have focused more on the question of whether there are perhaps hardwired genetic defects in airway smooth muscle itself. For example, mutations in contraction-inducing receptors or cytoskeletals or signaling proteins that may lead to a hyper-responsive airway smooth muscle mechanophenotype. And a number of our studies have focused on a large family of proteins called the regulators of G protein signaling. We know that G protein coupled receptors, such as those for leukotrienes or histamine, are the major controllers of bronchial smooth muscle tone. And these are a group of cytoplasmic proteins that negatively regulate the output of G protein coupled receptors. And we have shown that several of these proteins are expressed in airway smooth muscle. For example, null alleles in RGS5 lead to spontaneous airway hyperresponsiveness in mice. But more recently, we've turned to the question of allergens themselves. An emerging body of literature has suggested that allergens, especially those from fungi and those containing proteolytic activity, can exert pro-inflammatory effects on the lung by inducing epithelial activation and also barrier damage and production of cytokines. But their effects on other lung structural cells really have been unknown. And so we set out to answer a couple of questions. Number one, whether fungal allergen components are actually deposited in the bronchial submucosa in asthma and where they could directly contact airway smooth muscle. And secondly, whether these exert direct pathogenic effects on airway smooth muscle cells. And so we started with a fairly straightforward experiment. We used commercial secreted filtrate extracts of Aspergillus fumigatus that are used for skin testing and immunotherapy, and we treated human airway smooth muscle cells in culture. So as you can see, the confluent cells are long and spindle-shaped, and application of these extracts induce a prominent morphological change, clustering and rounding of the cells, and this was completely abrogated by either heat treatment of the extracts or by inhibition with a serine protease inhibitor, but not one of cysteine proteases. So our hypothesis became that there is a serine protease activity in these extracts that was affecting the morphology of these cells. And we also hypothesized that perhaps this protease is digesting extracellular matrix and affecting the interactions of these cells with their ECM substrate. And we saw this at the ultrastructural level. So we saw internalization of integrins, which of course mediate interactions with the ECM. And we also saw a decrease in number and size of these punctate structures, as shown by the arrows here, which are called focal adhesions. And this is detected in green by staining with the focal adhesion component vinculin. And of course, focal adhesions are multi-protein force-bearing complexes that cells use to attach to ECM substrates. And there are many of these, many components within focal adhesions that directly affect airway smooth muscle contraction. We then went on to isolate a serine protease that was present in these extracts. We used a serine protease inhibitor column and then mass spectrometry to determine that the major protease in these extracts was this 33 kilodalton protein called alkaline protease 1, or ALP1. And this is a known major allergen of Aspergillus called AspF13. And we found that the addition of the ALP1 protease, purified protease, could recapitulate the effects of the whole extracts on these cells, induce the clustering and rounding of the cells. This was, again, inhibited by a serine protease inhibitor, but not one of cysteine proteases, suggesting that this protease was mediating the effects of the Aspergillus on these cells. So because this was affecting the ECM interactions, something that's known to affect contraction, we next went on to ask whether this affected the contraction of these cells. And just to point out that this protease has a number of homologues that are present in many allergenic molds, including penicillium and mucor, for example, and Candida. So we think this has broad implications for fungal allergy. And so to study its effect on airway contraction in an ex vivo assay, we used the precision cut lung slice technique. And we took whole lungs from naive mice and then solidified them in agarose and make thin sections. And then you can visualize airway dynamics in response to bronchoconstrictors directly by microscopy and then quantify this. And what we found was that, again, these are naive mice that had not been challenged with fungi or had any kind of pre-existing inflammation, that pre-incubation of these slices with this serine protease could enhance the response to bronchoconstrictors, as you can see here, in a dose-dependent manner. And so we think that this has a direct impact on bronchial contraction. So we wanted to isolate a molecular defect underlying this response, and we reviewed the contraction signaling pathway. Airway smooth muscle cells contract in response to acetylcholine or its analogs carbachol, as I showed in the previous slide, by activating M3 muscarinic receptors. Most of these bronchoconstrictor receptors activate the cytoplasmic protein G-alpha-Q, and this will induce intracellular calcium mobilization and activation of the small GTPase Rho A. And in tandem, these will increase the phosphorylation of myosin light chain, and that promotes its interactions with actin filaments and leads to cell shortening and forced generation. And what we found was that this ALP1 protease affected both of these pathways. It increased the basal calcium levels, and while it didn't induce an acute calcium response in and of itself, it enhanced the peak response to bronchoconstrictors, in this case bradykinin, and this is a fluorescent measurement of using a calcium indicator of the fluorescence over time, and it enhanced the peak calcium response of these. Again, these are human airway smooth muscle cells. In addition, it also affected the Rho A pathway, so we saw increased bradykinin-induced Rho A activation in these ALP1 pretreated cells. So while the protease didn't really induce a signaling pathway that we can detect, it did enhance the responses to GPCR bronchoconstrictors. And we think that both of these pathways are important in promoting this hypercontractile phenotype. And more recently, we've used a biophysical assay developed by our collaborator Ramakrishnan at Harvard and others, in which cells, human airway smooth muscle cells, are plated in 96-well plates over a layer of ECM-coated beads, in this case collagen, these are green, and then underlying that is a layer of polyacrylamide embedded with red beads. And so using Fourier transform traction microscopy, one can quantify the displacement of the green beads, which again is mediated by focal adhesion interactions. And then more red signal is exposed in the image, and so the more force per unit area or contractile force that these cells have, you can see more red in the image. And I think you can appreciate that compared to the control cells, we see a lot more contractile force in the ALP1 pretreated cells. This is quantified here on the graph on the right. And then in both cases, this response was sensitive to pretreatment with a Rho kinase inhibitor, suggesting that the Rho A pathway was important in mediating these effects. So to turn to the second question, most of these were in vivo or ex vivo assays, so what relevance does this have to human asthma? So to study this, we first turned to our mouse model, and in our early studies, we used a conventional model in which mice are sensitized intraperitoneally with Aspergillus extracts and adjuvant, and then they're challenged intranasally with the Aspergillus extracts. And we examined whether this ALP1 protease was present in the lung tissue of these mice. We generated a polyclonal ALP1 antibody, and as you can see here, we saw in both acute and chronic challenge models the presence of this DAB staining indicative of the ALP1 protease in the bronchial submucosa in the areas that you would see airway smooth muscle, suggesting that this protease is present at least in these mouse models. In contrast, when we just gave the protease either for short periods of time, 4 hours or overnight, we really didn't see any of this protease present in the tissue. And so this has been a central question of whether this is a combination of pre-existing inflammation, perhaps there's epithelial damage and penetration of this protease into the lung tissue. And it's been a challenge to really understand what mechanisms are present that allow this protease to get into the tissue. And we extended these findings to human asthma and collaborated with Michelle LaViolette in Canada to look at ALP1 immunostaining of lung biopsies from a cohort of patients with asthma of varying severities. And you can see by the brown staining in the areas indicated by the red arrows, this is airway smooth muscle here. We also see it in the epithelium, but we do see it in the airway smooth muscle areas, and the amount of staining was directly proportional to the severity of disease in this cohort. And this is just quantified here in this graph. So there was a significant increase in the ALP1 immunoreactivity in the severe patients compared to mild to moderate disease and controls. And this had a direct correlation with lung functional impairment. On the graph on the right, we're comparing the amount of ALP1 immunoreactivity in these airway submuscle areas in the lung biopsies with the forced expiratory volume in 1 second, or FEV1. This is a measure, a lung spirometry measure of lung obstruction. So the lower the FEV1, the more severe disease. And I think you can appreciate that there is a direct correlation between the degree of lung obstruction and amount of ALP1 in the lung tissue. So as I mentioned, the challenge really has been that we've established an in vitro mechanism whereby this ALP1 protease can increase airway submuscle contraction, and there also appears to be a requirement for lung inflammation to allow this protease to get into the lung tissue. So how can we segregate or differentiate these processes in vivo? And so we've turned to a more physiological model of challenge of mice, just intranasal repetitive challenge over several weeks with Aspergillus extracts, or actually purified Alp1, and then examine these various mechanisms. And we found that we need about 1/10 of the amount of protein of the purified protease in a protease assay to be using about the same amount of protease activity as we see in the whole extracts. And so in this model, we see that the Aspergillus, and this has been described by others, the Aspergillus challenge will induce an airway hyper-responsiveness in these mice. This is a plethysmography response to the bronchoconstrictor methacholine. And what we do here is to use a tracheal catheter and mechanically ventilate anesthetized mice. And then an oscillatory waveform is applied through the catheter, and the instrument can detect changes in pressure and flow, whereby we can derive airway resistance as a measurement of airway obstruction. And you can clearly see that challenge with Aspergillus induces an increase in airway hyperresponsiveness in these mice. And what we found was that any heat inactivation of these extracts or pretreatment of the extract with a serine protease inhibitor completely abrogated this response. And we also found that ALP1 challenge of these mice in the same model with this ALP1 protease is sufficient to induce basically an equivalent response. And again, this is eliminated by heat inactivation of the extract. And what's been challenging is that this Aspergillus-induced airway hyperresponsiveness is partially uncoupled to the inflammatory response. So in challenging mice with heat inactivated extracts, we see essentially no airway hyperresponsiveness, and yet we still see an inflammatory response. We see generation of allergen-specific IgE, although there's a trend for a decreased response. And then looking at the lung histologically, we see by H&E staining there's still a variable degree of eosinophilic infiltration around the airways. And by periodic acid shift staining of the epithelium, this dark pink staining, which is a measure of mucus production or mucin staining, we still nonetheless see an inflammatory response and mucus staining of the epithelium even in the mice that were challenged with the heat inactivated extracts. And so this suggests that there's an uncoupling of this inflammatory response, and perhaps that ALP1 is contributing to the increased bronchoconstrictor response of airway submuscle through inflammation-independent mechanisms. And so because of this challenge, we've begun to look at several models of mice, of immunodeficient mice, you might say, that have impaired inflammatory responses to see if this mechanism is still present. And as a first step, we used eosinophil deficient mice. These are delta double GATA mice. They have a missense mutation in the GATA1 transcription factor promoter, and fortuitously it was found that they lack eosinophils. And so these mice basically had a response in terms of airway hyperresponsiveness to methacholine that was essentially equivalent to wild-type mice, and yet they had basically no eosinophilic infiltration of the airways. They did have some neutrophils, so perhaps these are compensatory, and they still have some mucus production in the epithelium, so perhaps these mechanisms are still operating in these mice. So this is not a perfect model to study the relative contribution of inflammation to this response. And we also looked at mice that were deficient in the protease-activated receptor PAR2, and this receptor has been implicated in the response of epithelium to a number of fungal allergens. And basically, we saw that the response of PAR2 deficient mice was essentially equivalent to wild-type mice, both in the amount of airway hyper-responsiveness that we see and also in the inflammatory response. One final note that what was notable in these mice was in these PAR2 deficient mice was that there was a lack of cytokine response. So IL-5 and 13 were much lower than the wild-type mice. And so this suggests perhaps that ALP1 is contributing to the airway hyperresponsiveness independent of type 2 cytokines. And more recently, we've tried to understand where these cytokines are coming from by immunophenotyping lung cells in this model. We find that there's a significant increase in the number of cytokine-producing cells in terms of IL-5 and 13 that are ILC2s. We don't really see a big increase in CD4 Th2 cells, at least at the time points that we're looking at. And so I think we're going to probably need to look at mice that are even more immunocompromised and basically lack adaptive immune responses, RAG, IL-2 receptor common gamma chain knockout mice, for example, that lack ILC2s, to better understand the relative contributions of the protease on airway smooth muscle function and the inflammatory response. And so just to summarize, we think we've identified a unique pathogenic mechanism, essentially the opposite of invasive fungal infection, where a lot of these patients with asthma will have airway mycosis. That is really not invasive infection, but there is a degree of microinflammation, if you will, in which the respiratory epithelium, the barrier function is impaired, which could allow penetration of this fungal protease and perhaps other components, we don't know, that has direct effects on the extracellular matrix interactions with airway smooth muscle via its protease activity and can thereby increase airway smooth muscle contraction and lead to airway hyperresponsiveness. And I'll just acknowledge the contribution of several of my collaborators, Narman Balenga, who's a postdoc who is now at University of Maryland, who had the original idea for this, and then more recently, several students, Tricia Basu, Jamie Reedus, and Morgan McCullough, who are really wonderful students who've advanced this project. Ray Panettiere at Rutgers, who's provided over a long period of time a lot of reagents and expertise, and then I've mentioned some of my other colleagues who made critical contributions to this work. So I'll stop there and take questions. Thank you.
Very nice work, Kirk. And again, because the microphone is right in my face, maybe I'll start out with a question. So your work with the precision cut lung slices is particularly interesting. So presumably there's no actual inflammation going on. There's no IL-13 and that kind of thing. So there's some sort of, that work begs the concept that there must be a ligand that engages a G protein coupled receptor that might be mediating that direct contractile event. I'm wondering if you have any idea what that might be.
Well, so I didn't present all the data, but we do see that even the baseline airway area in those slices is decreased if you pretreat with this protease. And so it's always been a question of whether you're simply digesting, and we know that the collagen content overall of those slices is lower after the treatment with the protease. So the question has been, are you really just having, it's like a COPD type effect where you're just digesting tissue, and so there's less tethering effects on the airways. Mm-hmm. But nonetheless, we do think it's an additional mechanism, because if we just add something like potassium chloride, for example, that will contract airways through changes in membrane conductance, we really don't see any effect. So it seems to be an effect on the GPCR ligand that's through affecting the whole signaling apparatus. And I didn't really describe fully, but we did identify mechanisms by which the Rho pathway was being activated, and also membrane phospholipid metabolism that I didn't really have time to talk about. But we do think it's really it's an effect on the response to the GPCR agonist. So this is any GPCR agonist that you add probably will have the same response. I know that was a long-winded answer, but okay. Does that make sense?
Thank you.
I have a couple questions. One is, have you had a chance to look at tight junctions directly?
We have not. So we haven't. One of the things that we found was we did early on. We kind of did a gene array type thing, and just to see what the effects of this protease on the airway muscle cells. And one of the prominent things that we saw was a decrease in the expression of N-cadherin, which is a junctional protein in airway smooth muscle. We haven't looked at the epithelial tight junctions, but in ASM, N-cadherin is decreased transcriptionally, and it's also decreased at the cell surface. And there's really nothing known about cadherins in airway smooth muscle function and asthma, and so that's an area that we'd like to pursue.
And the other question I had was, you showed that the serine protease inhibitor action on the extract reduced the result. But what about treating the animal?
So we haven't done those experiments. There already are a lot of, well, I shouldn't say a lot, a few papers in the literature in which you can use pre-treat mice with these serine protease inhibitors in an OVA model, I believe, and it's actually very effective. So the other avenue is to do some high-throughput screening. Perhaps we can identify a pathogen-specific protease inhibitor that might be of therapeutic, or an antibody perhaps against this protease that might be therapeutically effective.
David, do you have a question?
So in the patients that we treat with fungal asthma, often they improve their coughing and they feel better. They feel stronger and more confident. But it doesn't always change their lung function parameters very much. Their FEV1s may be similar, peak flow might be just marginally changed. Some of that could be because they just get rid of some mucus, so there's less mucus there. But I'm just wondering about the translation here, and David understands this very well, and I'm just wondering, therefore, the translation of this into the patients in terms of lung function parameters, because it has regulatory implications. So if you were going to try and register a new drug, the lung people at the FDA like to see changes in hyper-responsiveness, but that might or might not be what you see clinically.
Right. So basically you're concerned that changing the burden in and of itself of fungus is not going to translate into immediate effects on lung function or clinical symptoms. Yeah, I don't have a good answer. I'm sorry? Yeah, I don't know. I think this is I don't really have a good answer for that. This is a field that's the effects of these fungi on lung structural cells, as I said, even respiratory epithelium is such, it's an understudied area, so I really don't know at this point whether reducing fungal burden is going to translate into an immediate impact on clinical symptoms. I can say, though, and I think we were at the fungal workshop and someone asked, well, we give these patients with fungal asthma already a lot of bronchodilators. I think one important thing to think about in just treating asthma in general is that we really have no therapeutics that target the contraction pathway. It's all focused on bronchodilation. And so this would be a way to really reduce airway smooth muscle contraction. And that's that's my best guess at this point, that it might translate into more pronounced effects over time.
Maybe if I could follow up just a bit, because it's such an interesting and important topic. We see the same kind of thing, David, that you mentioned. So the antifungals work quite well in making people feel better, but the actual improvement in lung function is scarce to non-existent. I think airway hyperactivity, my feeling, Kirk, I'd be interested in your opinion. My thinking is that airway hyperactivity is something that's very important in early stage disease. But in the folks that I see, they're often in their 60s, 70s, and 80s that have been grappling with fungal asthma for many, many years. That they've moved beyond that. That there's structural changes of the airway, including bronchiectasis in particular, but airway fibrosis. I think you mentioned a little bit about that.
Yeah, I think
that complicate the situation.
Yeah, I agree with you. I mean, because you're entering COPD overlap territory, so it may actually targeting contraction. Those patients might not be as effective. Yeah, I totally agree.
Anyway. Other questions? All right, very good. Well, thank you.